Can you explain how certain medications can interfere with the LRA tests?
Yes, the LRA test, the lymphocyte response assay as we developed it, depends upon the membrane, the surface of the white cell, the lymphocyte, and the other white cells that are present in the blood being fluid. That is, the membrane is able to move around so that it can interact and detect and respond to foreign antigens. Now, there are certain classes of medicines that we know that decrease the fluidity of the membrane, and we know how long that occurs, and during that influence, you can get false negative results. So, with regard to some medicines, we need a 4-day avoidance because that's how long it takes for the active medicine to get metabolized and the effect on the lymphocyte membrane to diminish to the point where it's not significant. In a perfect world, we would have perfect people. That's not the world we live in. But over the 30+ years that we've standardized the test, we have found it really important to follow certain preparation rules, including avoiding steroids and steroid-like molecules for 4 days, and aspirin-like molecules for 2 days. However, many medications that do affect the immune system have no influence on the lymphocyte membrane. So it may seem paradoxical, but the only concern we have is that the blood be able to respond ex vivo in our laboratory. That is, the blood will respond just as it would inside your body. That's what ex vivo means, but in our laboratory. And in order to do that, the cells must be able to react, interact, communicate. Antigen-presenting cells, APC cells, have to wiggle over and present the antigen that is in that specific well of the microtiter plate where the incubation is being done. And after wiggling over, it has to present that processed antigen to what's called the MHC locus, the major histocompatibility site on the white blood cell lymphocyte. And then, within just a matter of hours, you see the first changes that, if you waited 3 weeks, would produce a colony of cells. If you use radioactive thymidine over 3 or 4 days, you could see the incorporation of new DNA being made. But we look upstream at an even earlier step that is the trigger that causes mitosis or cell division to occur or activation to occur. And so we actually look at a very specific kinase, and the consequences of that are the results of the LRA test. And as long as the membranes are fluid, we can do a good job. And that's why we have the specific preparation that we have.